Hi everyone. So welcome to our talk today. The title is Acquisition and Analysis of 3D Whole Body Skin Images for Dermatology Studies. And our lab is Medical Image Analysis Lab from SFU. And our group members are Zhao, who's undergraduate, Kumar, PhD candidate, Priyanka, former master internship student, me, Mengliu Zhao, and Jeremy, our postdoc to research associates, and Gasan Hamarne is our PI. So the reason we do this study is because skin conditions is a really serious problem. So it's one of the most common new reasons for people going to GPs in England. And 1 out of 4 Americans have skin diseases. And it's one of the most common reasons for people to go to GPs, while GP has less accuracy in diagnosis than dermatologists. But for visiting dermatologists, you have to wait extremely longer time. So on the other hand side, we see a huge increase in utilizing the AI methods these days. But state-of-the-art AI methods focus on 2D images only. But on the other hand side, the reason we utilize 3D imaging is because we assume it's imaging the whole body can offer additional context for diagnosis and longitudinal tracking. But then thinking of 3D imaging, we have to think about accessibility, portability, price, and quality. So these are the major concerns. In this work, our key contribution is we provide a novel pipeline of 3D imaging acquisition and AI-facilitated analysis of skins. So on the left-hand side, we can see an image. Sajjad is scanning the 3D mannequin. And we build up this 3D mannequin mesh. And we can see here in the middle, it is a zoomed-in look at the 3D version of the skin lesion. So what we did is for analysis, we unwrap the 3D mesh onto the 2D domain by checking the texture images, and we perform deep learning to the 2D texture images for localization. And we can see here on the right, the yellow bounding box is showing the detected lesion on the 3D mesh. And we also perform, excuse me, we also perform the matching on the 3D surface as well. So another key contribution of our work is we built these two datasets. One is the FAUST 900 and one is the EVA 420. So because of the COVID-19 stuff, we couldn't actually find human candidates for scanning. So that's why we built this synthetic dataset that we adapted the original FAUST dataset, which is colorless 3D mesh dataset, and we added synthetic skin colors, which follows the Fitzpatrick scale, and we augmented the original 300 3D meshes into 900 meshes with varying poses, and to each mesh, we add a single realistic lesion, which is sampled from the ISIC 2016 dataset. And also, Sajjad scanned a single mannequin, 3D mannequin. And we produced this 420 meshes, which is collected from the single mannequin with different postures. And for each mesh, there are multiple synthetic lesions. Then another contribution of our work is we perform this faster RCN on the texture images of those 3D meshes. So for the fast RCN, it's a quite mature deep learning network for detecting purposes. It's pre-trained on COCO dataset, fine-tuned on the synthetic FAUST 900 dataset we built. And we see a promising result that when we do the transfer learning, we test it on the EVA dataset, we can see that the performance is actually very good. So we can see on the right-hand side that we have this we have this detection result on the EVA scan texture images. So the green bounding boxes are the ground truth, and the red bounding boxes are the detected lesions. So we can see they are almost overlapping. So which means the qualitative result is really good. But we haven't done a quantitative result on the EVA scans yet. And on the other hand side, we did the matching for longitudinal tracking of lesions. We performed matching algorithm. So we can see, so we perform two kinds of matching algorithm. One is Hungarian matching using first order information, and then one is adapted from the PSL matching using higher order information. And we can see a matching result on the image below. So, a quick summary. Our work is proposing a novel pipeline of 3D image acquisition and AI-facilitated analysis. We build synthetic FALS 900 dataset with varying postures, realistic lesions, random skin colors, and physical scan EVA 400 dataset. We performed automatic lesion detection using fast RCN, and we perform automatic PSL lesion checking using higher order information on 3D meshes. And our future work will include obtaining more and better data, especially from real human candidates. And we hope to analyze depth and distortion of 3D scanning and extend our work to automatic diagnosis and other skin-related diseases. Thank you very much.